Before I share my data today, I'd like to thank the organizers for the opportunity to be here, both to share my data with you and for sponsoring my attendance at the conference. My name is Kristin Anderson, and I'm a postdoc in the lab of Phil Greenberg, and we're based out of the University of Washington and the Fred Hutchinson Cancer Research Center. And our major goal in the lab is to genetically engineer T cells to have a T cell receptor that's high affinity that targets tumor antigens. And we intentionally pick proteins that are present in multiple kinds of tumors so that we can develop tools that are broadly applicable for lots of kinds of cancer. So when I joined the lab, my goal was to use these tools that we'd already developed to work on treatments for ovarian cancer. And we initially picked high-grade serous ovarian cancer for several reasons. Not only is it one of the really aggressive types of ovarian cancer, most patients who present with ovarian cancer have high-grade serous, but in addition to that, the current standard of care often only results in a survival rate, a 5-year survival rate of less than 50%. And patients who present with really advanced stage disease, which is actually the vast majority of patients, have a really poor prognosis and a really high recurrence rate. So what we wanted to do was ask, could we use adoptive T cell therapy to treat patients with late stage disease? And in order to do this, what we wanted to do first was select a preclinical model that recapitulates as many hallmarks of the disease as possible so that we could basically test adoptive T cell therapy, identify any obstacles to the efficacy of that therapy, and then manipulate different engineering strategies and try combination therapies, anything we could do to overcome those hurdles and translate the most effective findings back into the patients. So the mouse model that we chose to start with is the IDEVEGF cell line because when we inject 5 million cancer cells intraperitoneally into B6 mice, so immunocompetent hosts, these tumor nodules develop throughout the peritoneal cavity. And they develop around 6 to 7 weeks, and these mice will eventually also develop terminal hemorrhagic ascites. So you can see an example in the ultrasound of some of these nodules that are growing along the pancreas, as well as early stage ascites developing. And so what we can do with these tumors is model advanced stage disease. Now, before we could dive in and actually start doing therapy, we had to ensure that this particular model recapitulated expression of the antigen that we wanted to target. And when I joined the lab, there were already a lot of tools that had been developed to target the protein mesothelin. So mesothelin is actually a cell surface protein that's expressed at low levels on mesothelial linings of organs like the lung and the heart. And what we realized is that it's also incredibly overexpressed in some really aggressive kinds of cancers, like pancreatic cancer, mesothelioma, and high-grade serous ovarian cancer. So you can see in the immunohistochemistry images that we have high brown staining in the human samples, and we also see this brown mesothelin staining throughout our IDEVEGF tumor samples. We also, when we look at the levels of expression in these tumors, find that relative to normal lung or normal ovary, we have a much greater level of expression of this protein in our tumors. So this made it a really nice antigen to try to target as a tumor antigen with our model. So in order to set this up, what we do is we isolate P14 splenocytes. So these are transgenic CD8 T cells that have a T cell receptor for the GP33 epitope of LCMV. And we activate them overnight with antibodies, anti-CD3 and CD28, in the presence of IL-2. And then on 2 consecutive days, we transduce with TCR-containing retrovirus, whichever T cell receptor we want to encode. And then we can actually enrich the population of T cells for the ones that were successfully transduced if we culture those cells with irradiated peptide-pulsed splenocytes. So once we have a nice pure population, we inject the T cells into mice that are bearing tumors, and we look at what happens. Now, in this particular experiment, it's important to note some ideas in this setup. Firstly, we wait until the tumor nodules in the peritoneal cavity are detectable. So we wait at least 6 to 7 weeks before starting therapy in order to model advanced stage disease. Because we know when we start seeing patients in the clinic, they're going to be at an advanced stage. So we wanted to try to test with a really high bar. In addition to this, these T cells that we are injecting are also congenically labeled. So the P14 cells we transfer have a Thi11 marker that we can detect and tell the difference between our donor cells and the recipient mice. And finally, we didn't do any lymphodepletion prior to this particular experiment because our question was simply, what happens if we put T cells in? So the answer is T cells do get in, they infiltrate the tumor, and what I'm showing you here is CD3 staining, so this is all T cells. But when we do enzymatic digestion and isolate the T cells from these tumors and do flow cytometry, these are predominantly Thi-1.1 donor cells within the tumor. So our donor cells, our engineered cells, can infiltrate tumors, but they wane very rapidly. So they don't persist long term. And that's problematic because for T cells to clear tumors, they have to be functional for a long time. So there are several reasons why this might be. And George Kukos's group actually published a couple of years ago a really nice suggestion or idea as to what might be happening. And his group showed very elegantly that in the blood vessels of both human and IDEVEGF blood vessels in the tumors, there's upregulation of Fas ligand, and that's a death ligand. So all our T cells upregulate Fas when they're activated, and this can actually induce CD8 T cells to die. So in our current iteration of T cells that we've made, these T cells are susceptible to this signal as they're entering tumors. So one way we are trying to get around this is actually by working with a fellow postdoc in the Greenberg lab, Shannon Oda, to make what we're calling immunomodulatory fusion proteins. So these are proteins that fuse the ectodomain of the Fas receptor with internal signaling domains from either CD28 or 41BB, some costim molecule, to try to take that negative death signal and turn it into either a survival or a proliferative signal or both. So we already have some data in vitro suggesting that when we co-culture these cells that contain these constructs in the presence of the ligand, we actually get enrichment of the cells that express these particular constructs. So currently, the in vivo experiments are ongoing, and we're really hopeful for what we will find. Now we know that this is not the only immunosuppressive pathway at play in our solid tumors. We know there are many pathways by which T cell dysfunction can be driven in solid tumors. And so we've done a lot of investigating to characterize what sorts of immunosuppressive mechanisms are at play in human high-grade serous ovarian cancer, because in order for us to investigate or explore strategies for overcoming those hurdles, it's important that we have a model that mimics all of those suppressive pathways. So we know from immunohistochemistry staining that there are Tregs in these tumors. We know that there are myeloid cells in these tumors. And so we are investigating various strategies that either deplete these cells or use molecules to try to convert suppressive cells from, for example, an M2 suppressive phenotype macrophage to an inflammatory M1-like macrophage to try to promote a T cell response as well. We also know, and I'm going to show you two examples here, that there are inhibitory ligands expressed in these tumors. So I'm showing you galectin-9 and galectin-3, which are proposed to be the ligands for TIM3 and LAG3 respectively, which are receptors that send inhibitory signals to T cells. And we know when we pull the T cells out of these tumors that our T cells are expressing these inhibitory receptors to some degree. So we're investigating whether or not blocking the signaling pathway can improve T cell efficacy and T cell function. But in addition to that, we're investigating whether or not, like before with the fusion proteins, you can usurp the signal and take that negative signal and turn it into something that actually promotes an anti-tumor T cell response. We also know that high-grade serous ovarian cancer is incredibly hypoxic. So there's a lot of regions within this tumor that are low in oxygen. And we know from the literature that that can be detrimental to T cell function. So in our mouse model, we also have seen that these tumors seem to be very hypoxic. So we're working with some companies and some collaborators to use small molecules that can actually deliver oxygen to these regions of low oxygen to try to promote T cell responses. And finally, we've also seen that these tumors are incredibly glycolytic. So seahorse data and data from the literature have shown that cell lines and tumor lines, lots of different ovarian cancer cells use glucose as a primary source for their metabolism. And so when we did a metabolite panel with collaborators at the University of Washington looking at what was present in these IDEVEGF tumors, we saw that glucose was very limited, which makes a lot of sense because if these tumors are rapidly using the glucose to grow, that doesn't leave a lot of glucose left in the tumor microenvironment. And this actually poses a potential problem to the T cells that we're engineering and hoping to have function in these tumors, because they rely on glucose as well. Data from Erica Pierce and others have very elegantly shown that effector T cells can become dysfunctional or even die if they are exposed to environments with very low glucose. So we're investigating whether or not there are ways we can promote these T cells using alternative metabolic pathways. If they can access pathways that use some of the metabolites or nutrients that are present in these tumors, we might be able to get around this obstacle as well. So my ultimate question that I'd like to share with you was whether or not adoptive T cell therapy targeting mesothelin could actually slow tumor growth in these mice. So for these experiments, what we tried to do was really put everything in our favor. We wanted to give T cells multiple times in the hopes that that would overcome this death or this lack of persistence. And we used a strategy that a fellow postdoc in the lab had optimized in a pancreatic cancer model where she had seen success in getting T cells to persist and actively prevent tumor growth and even start to cause tumor shrinkage. So what we did was we made T cells, as I described before, and again, we waited until late-stage disease where tumors were already detectable. And this time, prior to the very first T cell treatment, we did lymphodeplete with Cytoxin. And when we did give our T cells, we always gave them in combination with antigen-presenting cells or splenocytes that had been irradiated and pulsed with peptide. And we did that not once, but every 14 days for the entire life of the animal. And we took them down or euthanized them when tumor progression had gotten to a terminal endpoint. And what we found, importantly, was yes, indeed, when we do transfer T cell specific for the tumor, we can prolong survival significantly, more than when we give T cells that are irrelevant, and more than when we have tumor-only controls. And we do believe that this is actually due to tumor cell death, because when we look at cleaved caspase-3 staining within these tumors, the tumors that were treated with mesothelin-specific T cells have a significant upregulation of cleaved caspase-3. So we found this very encouraging. So if you take one thing away from this talk, it's that we do think that engineered T cell therapy can work in some of these solid tumor models, but that we have several hurdles and obstacles we'll want to continue to modify our cells to overcome in order to make this therapy effective when we translate it to the clinic. So with that, I would like to thank all the people who contributed to the work, the members of the Greenberg Lab, as well as our collaborators, and I would also like to thank my funding sources. Thank you.